In this video, we want to examine the neuromuscular junction, its anatomy, as well as its function. And what we have in this figure is the axon of a lower motor neuron coming down and innervating a muscle fiber at the bottom here at the sarcolemma, the muscle cell membrane. Now, these lower motor neurons originate in the ventral horn of the spinal cord or in the brain, and those axons would then run in cranial nerves. But whatever the target, those motor neurons tend to branch. We're only showing one of those branches here. And they branch and innervate quite a few individual muscle fibers, forming motor units that will work in unison. So they work as a functional unit. So let's just keep that in mind, even though we're only looking at one of these. Now, as a neuron action potential is generated here, we're going to get a nerve impulse, travels down to the synaptic bulbs here. We're going to depolarize that bulb with the arrival of that nerve impulse. That opens our voltage-gated calcium channels. Calcium will enter the synaptic bulb, and that initiates vesicle fusion. In this case, we're going to release acetylcholine as the neurotransmitter. So our acetylcholine is released into the synaptic cleft, shown here. It will diffuse across that tiny distance, in some cases finding and binding to our nicotinic cholinergic receptors. Right? So these are acetylcholine gated channels, and they're actually permeable to both sodium and potassium, which might sound contradictory, right? One ion would tend to come in, the other would tend to go out, might cancel each other out. However, the potassium is very close to its equilibrium when a muscle cell is at rest, and so there's really very little potassium flux. Sodium, on the other hand, is very far from its equilibrium, and therefore sodium flux dominates, and we get depolarizing end plate potential, which is essentially a graded potential. But in this case, we call it the end plate because it's sort of a specialized, very specialized synapse. It's very large, and we have these invaginations of the sarcolemmal membrane. And the rationale there is that we want a very large depolarization of the skeletal muscle fiber anytime we get a motor neuron action potential. And what that does is it effectively turns this synapse into a relay, such that we get an end plate potential that's large enough every time to generate electrotonic current that will open voltage-gated channels surrounding this synapse. And so now we have a positive feedback loop where voltage-gated channels will cause depolarization and open more voltage-gated channels, and an action potential will travel along this sarcolemma. It'll go down into the T-tubules and off to the ends of this cell in either direction here, right? Effectively generating the initial stimulus for excitation-contraction coupling, right? The generation of twitch tension, twitch meaning the tension generated with a single action potential in this skeletal muscle fiber. Now, even if we only got one action potential in our motor neuron and therefore one action potential in our muscle fiber, we would get twitch, but we could also rapidly turn this off. And that happens through the expression of an enzyme called acetylcholinesterase. And acetylcholinesterase, as the name indicates here, is an enzyme that degrades acetylcholine. So we very rapidly start to degrade these molecules of acetylcholine, right? We've got our enzymes embedded in the membrane here. That would, of course, reduce the acetylcholine binding to these gated channels, reducing, eliminating over time our end plate potential. Actually occurs quite rapidly. And then, of course, we would no longer be at the threshold of our voltage-gated channels, and our sarcolemmal action potential would end. Now, if we get another action potential that arrives immediately after this, right, we would get more acetylcholine, and we could effectively have twitch after twitch after twitch in the muscle fibers innervated by this motor neuron.